The EN17272 standard was released last year, and it describes the methods for testing airborne room disinfection by automated processes. As most disinfectant standards, it all starts with culturing the microorganism of interest in advance of testing, so to have a fresh culture ready. The first step on the day of the test is to prepare the microorganism carrier. These are stainless steel disc grade 301 2B surface finish of 4 cm diameter. The discs are soaked in 5% Deacon 90 for 60 minutes, followed by a washing step performed using sterile distilled water to remove any detergent, and ultimately soaked again in a bath of 70% isopropanol for 15 minutes. After soaking in isopropanol, the discs are allowed to dry under laminar flow or inside a Class 2 safety cabinet for as long as necessary. Test tubes and agar plates are labeled and poured and diluent dispensed into containers. Interfering substance, i.e. to mimic clean or dirty condition in real life, are also prepared and diluted to the correct concentration prior to be mixed with the test organism culture. At this point, a suspension of the microorganism is prepared. This is performed by harvesting surface culture using a sterile loop. Loopfuls of the cells are dislodged on the side of a 100 ml flask containing 10 ml of sterile diluent. The flask is then shaken for 3 minutes and the suspension standardized to achieve the required concentration using a spectrophotometer. The suspension is then mixed with the interfering substance prepared previously to create a final inoculum suspension ready to be inoculated onto the surface of our carriers. The inocular suspension concentration is confirmed by performing 10-fold serial dilutions down to the estimated concentration. The final dilutions are plated out by performing a 1 ml pour plate alongside a membrane filtration count. Once the carriers have completely dried, we are ready to inoculate them with 50 microliters of a final inoculum suspension, which should result in a minimum of 1 × 10⁶ CFU per carrier. The 50 microliters are aliquoted on top of the carriers and spread to a diameters of about 2 by 2 centimeters using a sterile loop. The carriers are left to dry inside a sterile empty petri dish for sufficient time at room temperature under laminar flow or inside a Class 2 safety cabinet. Once dried, the lid is closed and the carriers are transported inside the test chamber. Here, triplicate carriers for each test organism are clamped with the inoculum facing away from the release source at a standard distance at a height between 1 and 1.5 meters. Duplicate control carriers are left inside the petri dishes and transported to a chamber with similar environmental condition as per the test chamber. All of our test chambers can measure relative humidity, temperature, and hydrogen peroxide ppm. Once everything is set up and the initial required environmental parameters are achieved, the decontamination cycle can start. Once the decontamination process ended and it is safe to enter, the exposed and unexposed test carriers are placed inside a sterile petri dish and transferred inside a Class 2 safety cabinet. Here, the carriers are transferred into a 250 ml sterile flask containing 100 ml of recovering liquid medium. The carriers are then scraped off using a sterile pipette to dislodge all surviving cells into the recovering medium. The next step is to perform serial 10-fold dilution of the recovering medium containing the carriers, followed by duplicate 1 ml pour plate technique using appropriate agar medium such as tryptum sodium agar for each dilution. The remaining liquid is then screened for any surviving organism by using a membrane filtration system. Finally, the stainless steel carriers are placed with the inoculum facing up onto an empty petri dish and molten agar medium poured over it. All samples are then incubated at the appropriate temperature and time according to the organism being tested. After incubation, all plates are counted and the test carriers count is compared to the control carriers count in order to calculate the achieved log reduction. If the required log reduction is achieved on each organism of interest, the process can be deemed successful in claiming efficacy for airborne room disinfection by automated processes.